as we're going through, and again, you've heard a lot about the ASOs. The nice thing about these are relatively, there's a lot of experience in terms of using them. They've been used for other conditions. Some of us have used these for spinal muscular atrophy. They're not ridiculously expensive to be able to manufacture, and we can adapt them. So we can make these for various different things. So next slide. Anyway, we talked a little bit about this already. So again, this won't make a permanent change. Next slide. So the way to think about this, I used this slide this morning, but just to say very specifically, it's like having a pair of scissors and going in and sort of clipping or being able to disrupt one of the two copies of the gene. And again, we need that specificity in terms of doing that. Next slide. And as we do this, I won't go into this in tremendous detail, but I will say that in doing this strategy, it's not perfect, but I often say, don't let perfection be the enemy of the good. And the reason I say this is because having 50% of KIF1A probably is not what I would do if I had my druthers. But on the other hand, having 50% of good KIF1A is a much better thing than having 50% bad KIF1A. And so, again, one of the things that we've been wanting to do within the org is to look at people that have 50% of KIF1A to know what it is we're compromising in terms of doing this. And as we've looked at this, I don't think it's a bad compromise. I think it's actually a pretty good compromise. But the perfect thing would be to go in and fix it, just to edit it and just do that one specific thing and get to 100% of good KIF1A. Next slide. So we went through this a little bit in terms of the handle. So we'll keep going through this, and we can keep going through this. But let me get down to some of the specifics, just so that people can sort of visualize or think for themselves what this is like. So for any of, and I'm going to say women, but for any of the women who've had a baby and have gone through labor, some of us have had an epidural, if you can remember back. And so that was something that helped in terms of going through the labor pains. So they stuck a needle in our backs here to be able to deliver the medicine in a place where it was able to go around and to sort of numb things up. Dr. Bain, who's right up here, is the one who actually is the one who does this, but putting the needle into that same space, and there's fluid that goes from the back around the spinal cord and actually goes through the brain and bathes thing in cerebrospinal fluid. So you can be able to deliver drugs to the brain by essentially going through the back. That's a safer way and a more accessible way for us to do that, because we've got hard heads. We've got this skull up here, so it's not like we can, stick a needle right there. In doing this, though, you can imagine, and I want you to imagine your family member doing this, you have to be very still to do this, right? Because as you're doing this, you need to get in just the right place. You don't want to go too far. You don't want to not go far enough. And so in doing this, it's very typical for us, if someone can't sit still, if they're a wiggly worm, to be able to give them sedation in terms of being able to do this. And so sedation is something that when we need to do it, we do it, but it's also not something that we take lightly, right? Because there's always some risk with doing that. Okay, so in doing that, it takes a few minutes to be able to get the liquid in. We also take some liquid out so that we don't overflow the brain with liquid as we're doing it, but that's the procedure we do. The one other point that I make with that is that as we're doing this, if someone has, for instance, a rod in their back, so if they have scoliosis, if they've had surgery, if they have a metal rod in their back, this is, as you can imagine, poking it in, is not something that's as easy to do. Next slide. So in doing this, and I want to, be fully transparent in terms of doing this, we do this and we do a lot of things to make sure that this is safe. And so we're monitoring to make sure, for instance, that someone doesn't have a bleeding problem, because otherwise sticking a needle in the back, we don't want people bleeding into there. We don't want people bleeding into their back or their head. We need to make sure that the rest of their body's functioning well, that there won't be toxicity. And within this, there's always the problem. There have been other studies, and we'll answer any questions you like, but there have been other studies with ASOs where there have been toxicities. And so I want to be very clear about this. This is not that it's a risk-free procedure. Some of you know, because you've been watching the rare disease communities, there was a trial with Angelman syndrome, for instance, and they had the same type of strategy where they were sticking the medicine in here. And some of the individuals had what we call a neuropathy. So this was not that Angelman syndrome started out with a neuropathy, but there was irritation of the nerves that come out of the spinal cord with that injection, As they stopped it, it did reverse, but that actually stopped that whole clinical trial with Angelman syndrome. Okay. And they had to go back to the drawing board. They had to reformulate it. They had to rethink about doing the ASO. So again, it was reversible. That's the nice thing about the ASO, is that you can come back from that. But again, I'm just being completely transparent in terms of that. I'm not saying we've had that problem with KIF1A, completely different condition, completely different ASO, but there have been complications. There have also, with one of my colleagues, been even more serious complications, again, with a completely different disorder, a completely different ASO, but where it caused water on the brain or hydrocephalus. And so that's another serious type of complication that comes from this. So we don't take any of this lightly. We take it very, very seriously. Next slide. So in doing this, and I'm going to make a distinction, and I know it's going to be very confusing, but I'm going to make a distinction between things. When we think in a lot of the work that we do to try and get a new medication to come forward, we call this a clinical trial. And by doing this, it means that we're putting people through. We like to do this in a way where we call it double blind. And what that means in terms of this is that the person getting the medicine, they don't know whether they're getting medicine or basically a sugar pill or a placebo. And it could be literally a pill, or it could be a fluid or whatever. And when it's double blind, it also means that the person administering the treatment also doesn't know what they're getting. So you don't know, I don't know, neither of us know. The good thing about doing it that way is it leads us, neither of us, to be biased, right? And so otherwise, if you know what you're getting, if I know what you're getting, sometimes I want so badly for something to work, I can hope and I can maybe convince myself that I'm seeing something that's not really there. And so to keep us completely honest and to keep us unbiased, the most rigorous way of being able to see if something's working is to do that double-blinded procedure. Okay. So when we start talking about at some point in the future, not tomorrow, not next week, but at some point in the future, when we do this in a really rigorous way, I do think the agencies are going to have us do this, and they should, in a double-blinded way, right? Now, other people worry and they say something about, well, I wouldn't want to be in that placebo arm, though. There's no way I'd sign up for a study like this if I might be in the place where I'd get a sugar pill. So many times the way we design these studies is we say for everyone, there is a period where you need to, if you're going to sign up for this, realize that you might be randomly assigned to that placebo arm. But at the end of the study, when it's all over, we would have something like an open label extension. So if this is safe, and only if whatever it is safe, maybe we would all agree that at the end of that, you would have the opportunity to be on the medication, whatever it is, and we would both know, right? That way we've got built into it the rigor of being able to gather the data we need to understand whether it's working, yet we wouldn't exclude anyone from being able to get access, right? So that's generally the way we think about it in terms of being able to build this. So we build it scientifically in a sound way, but we make sure that everyone gets access to this. So I'm just telling you sort of what, in general, how we do this and the reason why we do this. And that's because we do want to make sure whatever it is we might come up with in terms of a treatment really is working, really is safe, and that everyone gets access to this. Okay, so that's what we do in general for clinical trials. As we're doing that, and back just one step, when we do that and actually get to a clinical trial, an agency, and I know the FDA more than I know the European agencies or others, they're also very, very careful. If they were to have us go through and do this, they would very rigorously make sure that we made sure whatever we were going to use were very safe. So they would make us do toxicity studies and animals, make sure that there were no problems with that before we would go forward. And it takes a while and it's somewhat expensive to be able to do that, but there's a very high sort of stringency in terms of doing that. Okay, so there's that. Now we're going to go to what I was talking about this morning, which is very different. So the N of 1, if you go to the next slide, the N of 1, and I'm calling it trial, but it's not really a trial in the same way. In fact, it's very, very different in my mind. So within this is not, and what we did with Susannah as an example was not double blind. Within this, I knew, the Rosens knew, the FDA knew, everyone knew in terms of doing this. And this was considered by the FDA to be compassionate use. And I went through to the FDA to a panel with them, and I made the argument because this is a condition that's degenerative, because we don't have any other treatment, because of the seriousness, you should allow us to be able to go through and go ahead and try this on one person, and as a compassionate way of doing this, because we don't have any other choice. Our back is up against the wall to do this. Because of that, they did not require us to have the same level of rigor in terms of toxicity. So we actually took a leap of faith. We did do some toxicity studies. We didn't go into it totally blind, but we didn't do it nearly as rigorously as we would do it if we were to do a full clinical trial and make sure that we had the safety. So there is a bigger risk when you go into these N of 1 types of situations, right? And so I just want to be very clear in terms of that. The risk is higher. I'm not saying they don't work sometimes, but the risk is higher as we do that. As the FDA does that for this particular way that they do it, they do consider it an N of 1, like literally 1 in terms of doing this. As we go forward, the FDA doesn't have a completely clear path in terms of where we go from an N of 1 to an N of 2 to an N of a few to an N of many. So I'm just being, again, completely transparent that I don't know exactly what they're going to say to us when we go to them and be able to say, this is the data that we have from our first person. What do you think now about trying it with some other people and how we go forward? And I think many of us in the room are committed to making sure we can go forward in the safest way possible and in the fastest way that's safe to go possible. But I don't know what they're going to say and how fast they're going to let us go forward. But that's the official sort of guidance that they have in this way. So as we go forward, and we could do this again for a different ASO. We could make up a different ASO that's targeting a different position and go through the same type of strategy. Next. So as we do this, it is not exactly the same in terms of, we do measure things in terms of finding the outcome, but we do have from the FDA a certain amount of latitude in terms of what we measure and how we know whether or not we're having an effect. We also have safety studies that are built into this as well. Next slide. So as we're going through, it also is something that because there's not a company that's sponsoring this, it's not as if, and I'm just literally, it's not like Roche signed up and is sponsoring this as a study. This is something that investigators end up covering the cost in terms of going forward with this. And so far, insurance companies, because these are not approved treatments, insurance companies, don't pick up the cost from this either. Next slide. So as we think about doing this, I just want to, put it out there. Some of you are going to feel completely comfortable with this. Some of you looking at your faces are saying, I don't know about this. I don't know that I'd want to be the first one to jump in the water with this one. Maybe I'd be the 20th one, but not the first one. But all of this in terms of the things that you'll need to consider. Okay, next slide. So as we're thinking about this, one of the things that I know people have asked about, because we have people from around the world, I think eventually we want to be able to make sure that people from around the world are going to have access. There are different regulatory agencies in different parts of the world. And I think what we're all committed to do in terms of doctors and scientists is make sure that we can collect the information and learn together as we're doing this. But we are likely going to have to deal with whether it's the FDA in the United States or the EMA in Europe or other regulatory agencies in Australia or Japan. There are going to be different agencies, and I don't pretend to be expert, not even in the FDA, and am I an expert, but I'm definitely not an expert in other parts of the world. We do appreciate, though, that people need to be close to home in terms of being able to do this. So as these things get bigger, we will build into this ways of being able to do this at multiple sites. We can't guarantee it's going to be in your backyard, 30 miles from your home, but we are very, very much cognizant that we need to be able to do this on larger scale. Okay. So with doing this, I'll just put it out there so it doesn't freak you out at some point. I have been thinking about, as we think about administration with the intrathecal, are we going to have to ultimately think about other ways of administration? And so there are other ways, for instance, that we deliver cancer chemotherapy for brain cancer. There are other ways that we can get things to the brain. And I'll just say very briefly, someday, maybe we will think about other ways of getting to the brain besides going through the back. Okay. To give you a sense of this, this is literally the protocol or the study visits that we had for Susannah and going through things. And as you can see, it's not just a one and done. So it goes through, you end up going in, we make sure that we're clear for the administration. We come into the hospital to actually stay for observation after the administration. And then a week later, we go through and we see what the effect was. So you can imagine that each time we do this, and right now we're on a schedule of doing this every 3 months. This is basically 2 weeks of your life, plus or minus 10 days, 2 weeks in terms of doing this. Every 3 months, rinse and repeat and go on forward. And so at least right now, that's just to get you in terms of the mindset, that's what that takes. Next slide. And you saw that already. And so I think that's it in terms of all of this. So anyway, that gives you, at least in terms of what's going on now with the ASOs. We'll talk about some other strategies as well, but this is the one that I think is the nearest term that some people are going to maybe think about. And again, if we get to other things, the wonderful thing is if we ever got to gene therapy, gene editing, there are potential for one and done. So like we aspire to get to that point at some time, but not anytime in the next year, for instance. Okay,